guys, welcome back to my channel. I'm so glad to see you all here. So since the last couple of weeks we have been talking about weighted gene co-expression network analysis, and today is going to be the last part of it where I demonstrate how to perform this analysis. For anyone who hasn't watched the previous video, I will highly recommend you to do that, as in the previous videos we have spoken about the idea behind this analysis, various steps involved, also talking about good practices, as well as going into the details of the workflow steps that are involved in this analysis. Just a quick recap on the workflow steps that we discussed in detail in the previous video. So we start with gene expression data, which could be from microarray or from RNA sequencing, and we construct a weighted correlation matrix. We perform additional steps to identify modules, and once we identify these modules, we can correlate them to external phenotypes or traits, or we can identify driver genes. So I want to demonstrate all these steps programmatically in R using the weighted gene co-expression network analysis R package. So the data that we are going to use today is an RNA sequencing data of peripheral blood mononuclear cells from a group of 34 individuals, half of which are COVID-19 infected and half of which are healthy. And we are utilizing weighted gene co-expression network analysis to answer a few questions. So the questions that we are asking today are: what are the genes or clusters of genes, that is modules, that are significantly associated with COVID-19 individuals? And what are the genes that are significantly associated with severe COVID-19 individuals? So these are our requirements for today's demonstration. I will be utilizing WGCNA package, DEC2, GEOquery, tidyverse, correlation level plot, and grid extra, which we'll be utilizing at various points in today's tutorial. So let us start by fetching the data first. So let's go to NCBI GEO and type in here the accession ID. So this is the accession ID that we're going to use today, that's associated with the data that we want to use. And I'm going to download the file that says raw counts. So I'm just going to click on HTTP and it'll open a dialog box and you can download the data. I'm not going to do it because I've already downloaded the data. So you can download it from right here. So let us start performing this analysis. So first we will load the libraries. Once the libraries are loaded, we will read in the data. The file that you just downloaded from the NCBI GEO, we'll read that into R. So providing the complete path to the file, and I'm just going to copy the name of the file. Now that the data is read, let us get the metadata. So we are going to use a package called GEOquery to get the metadata. So just saving the GSC accession ID into a variable, and now getting the expression and the metadata using the getGeo function. So this is the pheno data that we fetched from the NCBI GEO using the GEOquery package. So let's take a look at this pheno data. So we have various columns, that is metadata associated with these samples. So I'm just going to click here and open it in the RStudio, so just that I can get a glimpse of all the columns here. So we have various information on the names of the sample, the GEO accession IDs associated with the samples, and a bunch of other information. But typically I'm interested in the last couple of columns that has information on the days post symptom onset, disease state, gender, geographical location, which is fine, but severity of the COVID-19 case. So I'm interested in this information. So I'm just going to subset these columns. So this is column number 46. So I'm interested from column number 46 to column number 50, and the first and the second column. So I'm just going to subset this pheno data. I'm going to get column 1, column 2, column 46 to column 50. I'm going to run this. And now let's take a look at the pheno data again, just to make sure that we have all the relevant information. So now that we have downloaded our pheno data and subsetted it, let us take a look at our expression data now. So looking at the first 10 rows and the first 10 columns, we can see that the first column here are the ensemble gene IDs, and the following columns are the samples, and the values are the counts. So what I want to do is I want to make a few changes here. I want to change the sample names here to GEOquery. And if you notice, there are some differences in the way the sample names are represented in both these data frames. So here the sample names do not have the hyphens. So in order for me to match these sample names, I need to make sure that they match 100%. So I need to change these dots to the hyphens. So, and I will have to perform some data manipulation steps. So let us do that. So first I will need to gather all my samples in a column called samples and counts into a column called counts, and I'm going to exclude the first column, that is ensemble ID, and let us take a look at how that looks like. The value would be counts, and you can see that all our samples are in one column and the counts in the other. So now let us go ahead and change the sample names. So I will mutate samples and I will substitute the dot with hyphen, and once I do that, I need to do an inner join with the pheno data. So I need to merge both the data frames by a column that is samples here and title in the pheno data, because the samples are present in the column called title in pheno data, and the same samples are present in our data frame in the samples column. So I need to tell R which columns to merge both the data frames by. And now let us take a look at this. So ideally we would also have all the other phenotypic data, that is the other columns in the pheno data, in addition to the columns that we have. So now let us subset these only to keep the columns that we want. So I want column 1, that is the ensemble ID. I do not want this column, I want the column counts, and I want the GEOquery. So I want columns 1, 3 and 4. So let us subset these columns. Let's take a look at that. So I need these 3 columns, and now I'm going to change the shape of the data again. So I'm going to spread the data such that my columns would be GEOquery and the value here would be counts. So this is exactly how I wanted. I haven't changed anything here. I've just changed the names of the column names, that is the samples, and I've just changed the shape of the data. So now this is the exact format I want my data to be in. And now I will want my first column, that is the ensemble ID, to be the row names. So let us do that. Column to row names, and the name of the column is ensemble ID. And now let us assign it back to data variable. So now that we have downloaded the metadata and the expression data and manipulated the expression data, the next step is to perform quality control. And the goal of this step is to detect the outlier genes and samples and exclude them from the analysis. So one way to do that is by using a function called goodSampleGenes, which is a function from the WGCNA package. And it basically detects whether any genes or samples are outliers. So the input that it requires is an expression data frame. And the format it requires the data to be in is that the rows should be the samples and the columns should be the genes, which is the exact reverse of what we have, where we have the rows as genes and the columns as samples. So we just need to transpose our data. So we can use a function called T, which transposes the data and provide it with our data frame, and the output of which can be stored in a variable called GSG. So this GSG variable holds a list of objects. So I just want to show you what the results for GSG look like. So it basically holds logical vectors, and basically it just shows whether there are any genes or samples that are detected to be outliers. So if you want to evaluate whether there are any outliers detected in your data, then you just need to look at the all OK vector. If this is true, it means that all the samples and the genes have passed, so none of these genes or samples are detected as outliers. But since the results are false, it indicates that either or both the genes or the samples are detected as outliers, and we need to find out those genes and samples and exclude them from the analysis. So I'm just going to do a table and count the genes that are, or samples that are detected to be outliers. So it seems that around these many genes are detected to be outliers, and we need to exclude them from the analysis. And let us check the same for the samples, and it seems that all the samples have passed the filters, and it seems that all the samples are good to go. Again, you can change the filters and the threshold according to your requirement. I'm just going here with the defaults. So let us exclude the outlier genes from our data. Another method to detect outlier samples is to perform hierarchical clustering. And let us see if we find any outliers in our dataset. So I'll be utilizing a function called hclust, and again providing it with the transpose data, and the method we are going to use is average. And let us assign it to a variable called h3. Now let's plot this h3, and zooming in on the plot, we can see that there are 3 samples that are distant from the rest of the samples, and I would like to exclude these 3 samples from my analysis because I think including them might skew my results, and hence I would want to consider them as the outliers and exclude them from my analysis. Another method to detect outliers is to perform principal component analysis. So let us compute principal components for our expression data. So we can do that in R using a function called prcomp and provide it with a transposed expression data. And let's save the result to a variable called PCA. So now this PCA variable holds a bunch of information, and the information about the principal components is stored in a slot called X. So let us save this to another variable called PCA data. And just taking a look at the PCA data, here we have principal components computed for all the samples. Now let us also calculate the variance explained by each principal component. So we can do that by taking a square of the standard deviation. Let us get the percentage of the variance explained. And let's round this up to 2 digits. So for all the principal components, we have computed the variance, the percentage variance explained by each principal component. So let us create a plot now. Let us plot the first 2 principal components. We're just labeling the percentage of the variance explained by principal components 1 and 2 with the percentage values. And now let's look at this. So the error says that the data must be a data frame. So let us convert PCA data into a data frame. And zooming in on the plot, we can see that again these three samples seem to be clustering distinctly away from the rest of the samples. And with this PCA plot, we can see that these three samples seem to be the outlier, and hence we will be excluding these three samples from the further analysis. A quick note before moving on. So if your study design has a lot of replicates, please make sure that there are no batch effects present. In case if there are batch effects, then one has to correct for them, and there are various packages and methods available which can correct for batch effects. So please make sure that your data do not have batch effects before you move on with the rest of the analysis. Now let us go ahead and exclude the outlier samples from our data set. So samples to be excluded, I'm just creating a vector with the sample names. So again, looking at the plot, these are the three samples that I need to exclude. So just looking at the accession IDs, so GSM 4615000. Now let us exclude them from the data. And now you can see that we have excluded three samples. So our data subset has 31 columns, so 31 samples, because we've excluded three samples. Weighted gene co-expression network analysis requires the expression data to be in the normalized form. So you can use normalized microarray or normalized RNA sequencing data. So if you have FPKM or RPKM values in terms of RNA sequencing data, you can use them. You will just have to log transform them. Since the data we have today are raw counts, we will need to perform normalization. The author of this package recommends that we can use variance stabilizing transformation from the DESeq2 package. So we will be using that function to normalize our data today. So let us first start by creating a DESeq2 dataset. And the first thing we will need is a column data, or we can use pheno data. So since we have excluded the outliers from the expression dataset, we haven't excluded the outliers from the pheno data as well. So let us do that. So we need to filter out in samples to be excluded. So we basically need all the samples in the phenotypic data or in the pheno data except for these three samples. So now let us assign this to a new variable called call data. So now I want to fix the column names in call data. So as you can see that there are some additional characters and spaces in the column name. So I want to remove these additional characters and spaces from the names. So let us do that. I'm going to substitute the special characters with nothing. And let us take a look at the names again. So now we have gotten rid of these characters. Now let us replace these spaces. With an underscore. And now let's take a look at it again. And now that our names are cleaned up, so we don't have additional characters or spaces. So in order to create a DESeq2 dataset, we have to make sure that the row names in the call data and the column names in the expression data are identical and in the same order. So just making sure So the row names are present in the call names in data subset, so that is true. And we also have to make sure that they are in the same order. So that is also true. So they are in the same order. Now let us create a GEOquery dataset. So we have done this previously. So the count data will be data subset, call data will be call data. And for the design here, we are not specifying a model here because we need this GEOquery dataset to perform variance stabilizing transformation. So we are not specifying model here. So now let us remove genes with low counts. So essentially we want to retain all the genes that have greater than 15 counts in more than 75% of the samples. So we do this by calculating the row sums in greater than 15, in greater than 24 samples. So now we are left with these many genes which are having greater than 15 counts in more than 75% of the samples. Now let us perform variance stabilizing transformation. So we do it using a function called VST and provide it with this filtered GEOquery dataset. So to get normalized counts, we can type assay DDS norm function and just do a head. So these are the normalized expression values, and again to process it further using the functions in the WGCNA package, we require this expression data to be in the reverse format. So we want the rows to be the samples then and the columns to be the gene IDs. So let us just transform this and save it to a variable called normalized counts. Now we can use these normalized counts for further analysis using the WGCNA functions. So far we have read in the metadata and the expression data, the gene expression data. Then we perform quality control to detect the outliers and exclude the outliers, and followed by that we perform normalization for the gene expression data. Next we need to perform network construction, and the first step here is to choose a soft threshold. Now there is a function in WGCNA package that will help us to pick the right power that will help us build a network of scale-free topology. Now let us create a vector of powers for which the networks must be calculated. So we want powers 1 to 10 and from 12 to 50 with a step size of 2. So actually it's by. So when we look at the powers, we can see that we have various powers for which we need the networks to be calculated. So now let us call the function that calculates these networks at various powers. So the first value would be the normalized counts. The power vector would be power for all these powers. Then the network type is signed. We need a signed network, and we need the output once it is running. And let us store this in a variable called SFT. Now this has finished running, and we can use some metrics that is calculated by this function to decide which power gives a scale-free topology for our dataset. So the metrics stored within this variable is present in the fit indices slot. So let's save this into another variable called SFT data. So let us take a look at SFT data, And here you can see that for all the powers that we specified, there are a bunch of metrics that are calculated. So ideally we will be using two metrics here to decide the power that gives us a scale free topology, that is the R square values and the mean connectivity. So ideally we would want a power that gives the maximum R square and minimum mean connectivity. So let us visualize these metrics and visually pick a power that works best for our dataset and will give us a scale-free topology. So let us visualize the indices. So for the first plot, I want to create a plot for the R square values, just to get the names of the column. So I want to plot power on the x-axis and the R square values on the y-axis, and want to label by power. This is going to be our dot scatter plot. We want to put the label slightly above the points. We want to add a y-intercept at 0.8 and the color should be red. Let us name the x-axis as power, the y-axis as model fit. Let's make the theme classic. So let's save this to a variable and let's create another plot for mean connectivity. Let us save this to another plot, and now let's visualize these plots side by side. Actually, let's make this into rows, and now zooming in on the plot. Looking at these two plots, ideally you should be choosing a power that has an R square of greater than 0.8, that is above the red line, and has minimal mean connectivity. So looking at the plot here, we can see there are multiple powers having the R square greater than 0.8, but you should ideally be choosing a power that has a high R square but not excessively large. So for my analysis, I'm going to choose a power of 18. Because it has a high enough R square above 0.8, and at the same time it has low mean connectivity. So I'm going to go ahead and choose 18 as my soft threshold, but feel free to experiment with the other powers and compare your results and see how that affects your clustering and your downstream analysis. Now that we have picked out soft threshold, we need to perform various steps to identify modules. So we start by creating an adjacency matrix and applying the soft threshold to create a weighted correlation matrix. And using this weighted correlation matrix, we can calculate proximity measures, that is a topological overlap matrix, which can be used to perform hierarchical clustering. We have discussed all of this in great detail in the previous video. So if you have not heard these terms before, I will recommend you to watch that video first before trying to perform this analysis. After hierarchical clustering, modules that are similar or have a high correlation are merged together. And all these steps are performed by one function in the WGCNA package called the blockwise modules. So this blockwise modules function uses a two-level clustering. First, it uses a fast, computationally inexpensive and relatively crude method to pre-cluster genes into blocks of size that is determined by the user, and then it performs a full network analysis on each of these blocks separately. At the end, the modules who are very similar and have a high correlation are merged. So the advantage of this approach is that it's less computationally intensive and it speeds up calculations. So in order to run the blockwise modules function, we need the expression data to be in the numeric format. So converting the expression data into numeric, so we use sapply and convert the normalized counts to numeric, and followed by that we will set the soft power. We just assign the power that we decided that we are going to use to a variable, and followed by that I want to assign the correlation function to a temp variable, because I want WGCNA to use the correlation function from their package. So this is just to prevent an error for namespace that we get if we don't do these steps. So we need to assign the correlation function to the WGCNA's correlation function. So now we start writing the blockwise modules function and we start providing it with the parameters. So the first would be normalized counts. Followed by that we have a parameter called maximum block size. As we discussed how blockwise module function performs a fast and computationally inexpensive clustering on a block of genes of certain size specified by the user. So this is the parameter where you would specify how many genes would you want to be included in one block, basically setting the block size. So now the size of the block should be specified keeping in mind the memory your system has access to. So the default is 5000, which is appropriate for most modern desktops. A 4 gigs standard desktop or a laptop may handle up to 8000 to 10,000 genes. A 16 gigs workstation can handle up to 20,000 genes, and a 32 gigs workstation can handle up to 30,000 genes. So since I have access to 16 gigs of RAM in my system, I'm going to set up my block size as to 14,000 genes, because I want all the genes to be processed in one block instead of that being splitted into multiple blocks. Setting the max block size to 14,000. The DOM type will be signed. The power that we want to use is the soft power, that is 18, that we stored in the variable called soft power. Then merge cut height should be 0.25. So this is the threshold that we want to merge the similar modules. Next is numeric labels. I'm going to set this to false because I want the module eigengene labels to be the name of the colors instead of the numbers. I'm setting a random seed for reproducibility and verbose to 3. And now let's assign this to a variable and run this. The blockwise modules function has a lot of parameters, and I'm running this function on my dataset with majorly default values, but this default values might not be suitable for your dataset. So I encourage you to read the manual and the parameters for this function and tweak them according to your dataset to see what works best for and what gives you the best results. And now that this has finished running, let us reassign the correlation function to the original correlation function. Now let us get the module eigengene information from the variable that stored the output from the blockwise modules function. So in the bwnet variable, the module eigengene information is stored as MEs, and you can save this into another data frame called module eigengenes. And basically the module eigengene data frame would look like this, where we have samples as the row names, and for each sample the module eigengenes have been calculated. And since we set the numeric labels as false, the names of the module eigengenes are the names of the colors. Now if we want to know how many genes belong to each module, we can simply do a table and colors, And when you run this, you get the number of genes that are assigned to that particular module. We can visualize the clustering in a form of a dendrogram, and we can create a dendrogram using a function called plot dendro and colors. The first parameter is the distance matrix that is stored in the dendrogram in the BWNet variable, and we want to visualize genes with modules before and after merging. So we will be combining the module colors before and after merging. So we get the unmerged ones and then we get the merged module colors. Let us label them. Next, let us set some plotting parameters. And let's run this. Zooming in on the dendrogram. So as you can see from the plot that there are fewer colors in the merged compared to the unmerged, which indicates that there were a lot of similar modules in the unmerged which were merged together into one modules. So basically we will be using the merged modules to perform further analysis.